So I like to start with this slide because it really does frame our thinking about this whole area, which is, and it's one I know a lot of people here share, including both Nathan and Lee, but we like to think of people as in states. Hopefully most of the time you're in a healthy state or a wellness state, if you want to use that term. And on occasion, hopefully rare occasions, you will enter a disease state. And we know many things influence the state. It's your DNA, obviously, but also lots and lots of other things that you're exposed to, your so-called exposome. And we're not so great at this. People are working on various methods, including us. But we can certainly see the consequences of these by doing these molecular profiles I'll tell you about in a minute. But we do know, again, that intuitively pathogens, food, stress, exercise will all influence your health. And I would argue a goal of many of us together, and this is where the big data comes in, it will be to try and understand this in quantitative terms. I do believe in the not so distant future, people will be born with their genome sequenced, and then understanding in the presence of what kinds of exposures will lead to what kinds of health outcome in some probabilistic sense, and then modulating that by, say, your food and other kinds of environmental exposures. So I think most people realize that there's been a revolution in the sequencing technology that's really driven a lot of what we can do now, but I would argue there's other technologies out there as well, and I think the ISB is very good about thinking about this, and most of the rest of the world doesn't think about this as much, but actually there has been a revolution in mass spectrometry. And I would argue there's a new wave emerging that's just as powerful as these other two waves in this whole wearable space about being able to do continuous sensing for being able to make lots of measurements that will impact people's health. And so we're employing all three of these technologies with the idea of trying to do data-driven wellness, data-driven health. And so this is, as you may know, about 7 years ago, actually 7 years ago last month, we set up something we call personal omics profiling, where we now measure 14 different omes. Most of the major ones are listed here, but in addition to your genome, we make all these other measurements. Most are out of blood, although metabolomics and lipidomics will do out of both plasma and out of urine. And of course, the microbiome we get from different sites. We're mostly collecting nasal and gut microbiome. Well, we're collecting 5 samples, and we're mostly analyzing these 2. The others are predominantly on hold. And then we also, as I say, in the last 3 years or so, maybe 3.5 years, we've added on biosensors. And we profile people longitudinally in a fashion you'll see. So every time we sample someone, if you will, in a very liberal sense, we're making billions of measurements when we do their methylome and microbiome and such. And so we're getting very, very deep profiles. The genome we only do once, the rest we'll do in this longitudinal fashion. So why are we doing all this? Well, we're really trying to understand what it means to be healthy. What is a definition of health in a personal level and at a state that hasn't really been described before, and then what happens when you transition into disease. We're also trying to understand how the different omes relate to one another. We know intuitively that your genome and your epigenome must relate to your microbiome, and which in turn relates to your metabolome and your immunome. We know they're all related, but we don't know that equation, and I would argue for any system, if you know all the inputs, you should be able to figure out the outputs, and again, in some probabilistic fashion, and that really is the goal. We also are trying to understand how individual responses can be similar and how they're unique at a personal level, so that if we threw, say, a rhinovirus on this room, maybe half people would get infected with common symptoms like, runny nose, things like that, and then maybe half wouldn't get but some might get severely ill, some less so, and we'd like to understand the personal side, the individual side as well. And then perhaps most importantly, there's no particular order here, as you do a project like this, we are very interested in identifying factors that will influence people's health. And I guarantee we find a lot of this, some of which is incredibly, incredibly powerful, and I'll show you one vignette or two of this a little bit later. So we will see things that are happening at a very early stage. So as many of you may know, we started this project now 7 years ago as of last month, and we've been profiling me through now roughly 250 time points, microbiome even more, probably about 400 or 500 time points. And I've been through 10 viral infections, all common colds. I've also had Lyme disease. I had a big bike accident last summer. And so what we do is we'll do dense profiling, typically about every 2 to 3 months when I'm healthy, But when one of these adverse events come along, we'll do many more samples. So as an example, 9 months into the study, I got this respiratory syncytial virus infection, and each of these numbers is a day. This actually ends about 2 years ago, but you can see as soon as I get congested, we'll take blood that day, next day, 2 days later, 2 days, so on. Dense profiling during adverse events, and then again, every 2 to 3 months when healthy, interesting times like before and after Malaysia, things like that we'll profile as well. You may notice there's a huge number of measurements down here. I've been doing various dietary perturbations as well to see the effect of different foods on my molecular physiology. So I'll go through this quickly, but because many of you may have heard the story before, but basically from my genome sequence, we predicted many things, most of which I knew already from my family history or from just general measurements. That is to say, I'm high cholesterol. I know I'm a great responder to statins. I knew that sort of thing already. That shows up. This is my complex disease risk profile, and they call these riskograms, where increased risk is the orange bar to the right, and blue to the left is a decreased risk. And again, mine made sense from my family history, all the ones with stars I knew about. Like nobody in my family has ever been overweight going back to my great-grandparents. But there were some new surprises, one of which was my genome predicted I was at high risk for type 2 diabetes. And if I plot, plot me relative to everyone else on this riskogram, I'm way at the far end of the distribution. So I went from what would be, for a male, a 27% chance of getting type 2 diabetes in my lifetime, to being 47%, which is at the high end. And that was a surprise for what I knew about my family history. And again, many of you may know this, but because we were doing all this tracking, including standard medical tests, one of the things we were following was glucose, which then actually shot up quite high after an RSV infection. And we were actually measuring this closely starting around here because of my genome. And we were surprised it was high. Right? I went in shortly after this infection. I had already cleared. That's about 3 weeks later. Repeated the test, came out high again, this fasting glucose. A week later, measured again, and it's arbitrary, but hemoglobin A1C, that's my levels. When you hit 6.5, you're classified as diabetic. My first measurement here was 6.4, so it's right on the edge. 5 weeks later, measured again by my general practitioner, came out 6.7, classified as diabetic. And so I know when that was. That was April 11th, 2011, because up until then, I had the world's worst diet. I would have eaten all those cookies and things you put out in the afternoon, and I cut all that stuff out. I had been biking. I doubled my biking, and I started running, and that's about 8 months. This is not a few days. This is a long period. It took about 8 months to bring it back down to normal. So this made a big splash at the time because the first time someone used their genome to predict their risk for a disease and then got the disease, and in my case, it was manageable. So now one criticism of this is, Mike, you don't know, that might have gone away on its own. So it turns out we accidentally did the right control because about 4 years ago, we started scaling up to do 100 people. Well, it started at 60, it's enlarged higher. This is my glucose. This is that RSV period I showed you before. This is my hemoglobin A1C. So during that period, I'd stopped running, ironically, right around here. And as we were scaling up, we were still collecting all the data, but nobody's looking at it, including me. And so right around here, somebody looked at it and said, Mike, do you realize your hemoglobin A1C is 7.0 again? I said, no, I had no idea. So I sampled me a few times, started running. As I said, stopped right around there. Started running, and I brought it down to 5.7, but that's about almost 2 years ago, and it's been creeping up ever since. And I've been running more and more, and it still keeps creeping up. And in fact, now it's at 6.8 for my hemoglobin A1C. I'm diabetic again, or at least above the threshold. So the bottom line is there's no question I have a glucose misregulation problem. It's not just me. I have 2 brothers, 3 sisters. They're all my build or thinner, and they're all elevated hemoglobin A1C. They're all borderline diabetic before. For all I know, they've hit diabetic. I haven't asked them recently. But the point is there's no question we have a metabolic disorder. The easiest hypothesis for this is that I'm genetically predisposed, and then in my case, going back here, is that the viral infection's what's triggered it. So it's probably a combination of genetics and environment. That's the hypothesis. I know anecdotally, lots of other people have had similar experience. It's not real science. That's why we're collecting more of these data sets to see, and I think this is going to be the power. But again, there's no question I have a metabolic disorder, which I am trying to get under control, and if it doesn't work, I'll be on metformin pretty soon. Okay. So as I mentioned, we are now scaling this to 100 people, and we're doing to them exactly what we're doing to me. That is sampling them every 3 months or so. And then when an adverse condition comes along, like a viral infection or an immunization, we will sample them more frequently. We also will sample them at other times. So we've had 7 people do a colonoscopy, so we're sampling them during that period as well. So other interesting times to see the effect of these events on these folks. And we'll see what's general and what's specific, and we see certain things already. And then a number of our folks are running through different dietary perturbations, and I'll tell you about one of those in just a few minutes. So we are, again, seeing what happens when healthy and then through these various perturbations. And I should mention these days, roughly half of our folks are pre-diabetic, which we think is an interesting population to study, not only because I now have a newfound interest in type 2 diabetes, but also because if you think about it, these are people who will probably get worse through their lifetime because age is the #1 risk factor for type 2 diabetes. And we can try and see what kinds of events are associated with that. And in principle, they could get better, although we haven't seen much evidence of that so far. All right, so again, then sampling during these periods. So just to give you a sense of where we are, we've been, as I say, for some individuals, we've been doing it 4 years. We've added on a few people more recently, so some of these folks only have 1 or 2 measurements, but many of them have quite a few measurements, again, over multiple years with these very dense samplings. And for some individuals, we have a lot of the data's in. There's a big processing going on right now. So summer, you'll see a number of the data sets out there. I forgot to say that what's nice is all of our folks have agreed to make their data completely open without names and birthdays. So they're putting their genome sequences out there as well. So they'll be in the public domain. There are a few exceptions, but nearly all of these 100 and some odd folks are doing that. So it'll be, we think, a very, very powerful resource for the community. So I can't tell you about all the vignettes. You will see lots of very interesting things come from a study of this sort, many of which are very, very strongly health-related, as you might imagine. So I'll just walk you through a few that I think will complement some of the themes I've heard about at the meeting. The first is genome sequencing. What can you learn from a genome sequence? A lot of people say, Mike, you know, complex disease, that worked for you, but we can't really predict that most of the time. And, you know, what do you really get out of a genome sequence? There's a set of 59 genes. They're called the ACMG genes, for those of you who follow the space, that if you see them in a genome, you should report that information. If you see a mutation there, report them back to the individual. And so we've looked at that. The word on the street is that that's 1% of people have a mutation in those genes. We did, in fact, look through now over 60, but from the first 60 where we poured over their genomes, as detailed as we poured over mine, 11 have a mutation that is reportable back. Okay, so it's a minority of individuals, but I guarantee if you're one of these 11 individuals, it's a big deal. As an example, one of these individuals has a BRCA mutation. I think most of you probably know what BRCA is, puts you at high risk if you're a female for breast and ovarian cancer. This individual is a male. He is at high risk for breast cancer, so he will get screened for that. He also has 2 sisters, so there's this issue, what does he tell his family members? We can get to that later. But there are other interesting ones as well. Let's see, I can tell you all kinds of stories. This individual just turned diabetic, was borderline, now is diabetic. Turns out he has a mutation in a gene called a MODY mutation. This is not normal type 2 diabetes. The way you treat a type 2 diabetic is one way, metformin, other things. A MODY person is treated completely different with sulfonylureas. So the treatment's 100% different, and the way he'll respond to this is 100% different as well. So actually, there's no way he was lumped in with the type 2 diabetics, but it's pretty clear he's a different kind of diabetic. And I would argue this is one of the reasons you want this information. I can give you another example. There's quite a few here. 2 individuals in our study have had heart defects that have actually come up, been exposed through our analyses here. This is one individual, and this is, I think, a good example of the future. This is an individual, we had just sequenced his genome, and his father had a heart attack. And he said, Mike, can you look in my DNA to see if there's anything relevant there? And he knew his uncle had died at age 50 of something. It turns out of a heart attack. And there was another family member with a heart attack. So basically, this is running his family, asked us to look in his genome. We did, and we found an obvious mutation in a splicing thing, and it does affect his splicing. We know this mutation, which means that A, we think we found the cause of the mutation, B, he's got it, and he did a stress echo, and sure enough, he has some heart issues. So now you could argue the family history in combination with the genomics is really what pinned this one down. Obviously, the stress echo pinned it down even further. But the point out of all this is that this whole study made him aware of this, and I would argue this is the power of having your genome. Even if you're only one of these 11, again, it's a big deal to have this information. Every one of these will affect the test they get when they go to a doctor's office. These individuals are at high risk for paraganglioma. They will have a urine test to look for that, which they would not have done otherwise had we not sequenced their DNA. So I would argue this information is powerful. It's not always good news to hear, but it's better to hear it than not, we would argue. All right, a number of our individuals are doing a very interesting study. It's a weight gain, weight loss study. So we're looking at the effects of short-term weight gain and loss. We sample them at baseline. They eat an extra 1,000 calories a day for 30 days. If they're female, it's only 750. So they gain on average about 6.5 pounds. We sample them 7 days after that period, and then they go on a weight loss. And then we profile them here, 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 and they're part of our study, so we profile them out further. About half of them, oops, sorry, we threw out one because he didn't follow the protocol right. So anyway, we're 13 insulin resistant, 10 insulin sensitive healthy. Insulin resistance is a precursor for type 2 diabetes. So basically, what I can tell you is that even at baseline, before they gain weight, we can distinguish the insulin resistant from the sensitive folks, no matter what omics assay we use. This is the RNA-seq data. The insulin-resistant folks will generally be activated or higher expressed for oxidative phosphorylation gene, which in hindsight makes sense. These other types of genes, these are the MODY genes. This is a surprise. These platelet express genes actually show up in every metabolic perturbation we do. It actually showed up when my glucose went out of control. It showed up in lots of different very interesting scenarios every single time that shows up. And then there are other things that are more rich for the sensitive folks. Here's the metabolomics profile, same thing. We can always find molecules that will distinguish the resistant from the sensitive folks. This is a particular metabolite. Here's a set of metabolites. Each row is a metabolite. Each column is an individual, and this is a measure of their insulin resistance. It's a special assay called an SSPG. But the point out of all this, there are molecules that distinguish the resistant from the sensitive, and you can do regression analysis and find key ones. This was known already for diabetics. We show it's there for pre-diabetics. This one, interestingly enough, comes from your microbiome. So in fact, the microbiome does shift for insulin-sensitive and resistant folks as well. We can, again, find key molecules and microbes associated with this. One of the cool things you can do, and here's where the systems analysis steps in, you can look for the pathways that change. And I won't get into details of this, but we basically normalize all the data and then look for patterns across all the different data types in both individually and integrative fashions. And you look for patterns like this one here. These are all the molecules that change. In blue are the ones that are low, get high when people gain weight, go back to low. These are high, get low, go back to high, and so on. One of the top pathways that actually changes turns out to be your inflammatory response. When you gain weight, your inflammatory response goes up, even though it's your peripheral system. Turns out another pathway that goes up, probably the top pathway, is your hypertrophic cardiomyopathy pathway, which we know that heart attacks are associated with gaining weight. We think this is a molecular explanation for this. These genes are higher expressed when you gain weight, and they return when you get lower. We do have other sets of genes, by the way, that don't return right away, so there are some long-term lasting effects if you do these short-term weight gains. We know that individually, everybody is different. So everybody is unique and special, and not just to their mothers. So the bottom line is these are cytokines, 63 cytokines and growth factors. Given this is an immunology-enriched conference, well, you'll notice every color is a different person. High is outside, low is inside, and every person is different. So the blue person has a lot of this one. I think it's IL-7. light brown, that one, IP10, orange, so on and so forth. Here's another light brown. Everybody has different baselines of cytokines. We think some of this is genetic and some of it's epigenetic based on studies from us and others. And so we think that this actually reflects people's susceptibility to different diseases, again, based on their baseline immune profiles.
Do they change over time?
Some of them, many of them are the same, but some will change depending on the person, yeah. And of course, they change during the viral infections, as you might imagine. There they go ballistic. So it's a great question, though. And some of them we think change with age, actually, very interestingly. So this is your microbiome. It turns out this wasn't discovered by us, but mostly I would say from the Human Microbiome Project, where they've shown everybody's personal microbiome is different. We all have a unique microbiome. What we found is that if you do any of these perturbations, say a viral infection or weight gain, weight loss, your microbiome will shift. So again, if you do a perturbation, you get a viral infection, your microbiome shifts, but you still look more like you than the person sitting next to you. So we can do cluster analysis. This is the way we do this, is you cluster up. You can do this with either 16S or what's called shotgun sequencing. Each color here is a different person. They've all done this. This is the weight gain one. The same thing's true for the for the viral infection. People will cluster. Everybody has done the weight gain, weight loss, and these folks will cluster by person. The samples cluster by person, again, not by weight gain or weight loss. So what that means is that for short-term perturbations, your microbiome's pretty robust. And I think that's because our eating habits and such are probably pretty constant during these periods. So and our epigenome is certainly very stable, it turns out. So we think, again, that your microbiome is pretty robust to at least short-term perturbations. I'm not so sure that's true for long-term effects. The other area that we've added on more recently is, in fact, biosensors. So you may be aware, I've lost track, but in 2015, we know that there were over 500 health-related biosensors out there on the market. And that number is even higher now. So we've gone out and we've sampled about 30 of these different devices to see which ones seem to be most accurate or convenient, and we can record and collect data from and work with the manufacturers, because that's a big part of a study like this. The power of these devices is that many of them, you can get continuous measurements, so you're not just sampling people periodically, but you're sampling them all the time. And so we collect roughly about 250,000 measurements a day on every single person. So constantly measuring them. So we tested about 30 of these. I've settled on 8 of them that I use every day now. I used to use more. And they range from, a simple scale to radiation monitor. That sounds a little eclectic, and it is, although it turned out to be interesting at one time. But a smartwatch is pretty standard, and all of our participants are wearing a smartwatch. We all have them download the Moves app, which is a free app that measures steps. And as I say, I think I use most of these, and our folks use the smartwatch and the Moves app and a few other things you'll see in a minute. And they measure a wide range of things: steps, heart rate is a big one, skin temperature, stress response, sleep, your blood oxygen, so on and so forth. So you can measure many, many different physiological parameters. So we've been collecting data and then learning how to work with it, and I want to just walk you through a few simple concepts because it sets the scale for what we're going to talk about. But basically, this is my circadian pattern. So this is hours of the day. This is my sleep, so I sleep at night, wake during the day, occasional nap in there. This is my heart rate, low at night, high during the day, not surprised. Skin temperature, high at night. It's different from your core temperature. Skin temperature, high at night, goes down during the day, comes back up. So you start defining your personal baseline. We do that for me. We do it for our other participants. This is 43 people. Everybody has a different resting heart rate. Everybody has a different, it turns out, skin temperature, and everybody has a different baseline blood oxygen level. So we can measure all these things. You can also group people by their activity. So this is grouping people into 4 categories. There are different pattern algorithms you can use for this, but these are people who are relatively constant during the day, so they're asleep at night. Relatively constant during the day, go to sleep at night. These are morning people, they spike in the AM. These are people who are active in the morning, afternoon. morning, afternoon, evening, so you can see what pattern you might fall into if you have one of these devices. All right, so the reason we do all this is to define people's personal baseline, understand what their rhythm is, and then the next step is to start looking for deviations from that baseline. So it turns out there's a number, but one of the most striking ones that showed up on me, because I travel too much, is that your blood oxygen drops during airline flights. This is known already. It's known in the literature, it's not known to most people. So this is a relatively short flight. This is altitude in green. It's my blood oxygen, starts high, drops low. Most airplanes, pressurized cabins at 8,000 feet. And so presumably that's what hit there, comes back up. We now measure this because it turns out there's an app on your iPhone you can get that measures barometer. What was not so clear was that how much of it's low. Well, it turns out 70% of the flight, I'm at 96 or below, and the significance of that number will become clear in a minute. And 5% of the flight, I'm 90 or below. So that's pretty low. So why is this a big deal? Well, it's not just true for me, it's true for everyone measured. This is the first set of 18 people, all different ethnic groups. I guarantee if you're on a flight that goes high enough, 30,000 feet or higher, and it's long enough, your blood oxygen will drop. We see no exceptions to this. Okay, again, this is known already, not well known, but known. All right, so here is something we couldn't find in the literature. When the blood oxygen drops, it correlates with fatigue. So this is my fatigue level. So you can blindly classify yourself as either tired or alert, and then measure your blood oxygen. And in blue are the times I said I was tired. This is on flights. This is on the ground. When I'm 96 or below, I'm generally tired. And when I'm 96 or above, I'm alert. So 96 is kind of the threshold. You could do a reaction time test, which is to say that here's blood oxygen. Here's my reaction time. Gets much slower as your blood oxygen drops, your reaction time gets slower. So what we think this means is that when you get on an airline flight, when you're all flying home tonight, the air pressure will go down in the cabin. You will get tired. It's not because you're exhausted from all the wonderful science here. It's because the air pressure is low. It happens every time. So, yeah, okay. Could be vibration of the plane helps too, but I think certainly this is a major correlate because you can also correlate it on the ground. If you're a workaholic, all is not lost because it turns out that, at least this is for me, if the flight is 7 hours or longer, my blood oxygen starts coming back up. So towards the end of the flight, it will be actually pretty close to what it was on the ground. So if the flight's long enough, you will adapt. The last long flight I took, I also put it on a stewardess. She seemed high the whole flight. So I think actually it's possible pilots, you'll be happy to know this, pilots and stewardesses, we hope, are adaptive going into the flight. All right. So again, these are amusing things that might affect what you do, but here's some other very cool stuff related to health. So the next step is to look for deviations from the norm. So it turns out this is one flight I took flying to Norway, flew to Frankfurt, then from Frankfurt to Norway, short flight. Short flights, my blood oxygen usually does this. This is my average. Drops, not a huge amount. This is what happened on that one flight. The median, and the same with the mean, was about 90 for my blood oxygen. When I landed, it didn't come back up. It stayed low. Okay? Turns out my heart rate was high. This is plotting outlying measurements across 2 years. Looking across here, this is right. That first blue line is when I landed in Norway. Lots of high heart rate measurements. So when I landed, I could see my oxygen was very low. My heart rate was high. I knew 2 weeks earlier I'd been helping my brother put up fences in rural Massachusetts where 55% of ticks are Lyme infested. So actually, then I got a low-grade fever, suspected I had Lyme disease, went to a doctor there, and basically he took blood, said my monocytes are up, and said, yes, I had a bacterial infection, and he recommended penicillin. And I warned him it might be Lyme, but he still recommended penicillin. And I said, no, I think I need doxycycline. And as you might imagine, it was tense for a few moments there. He did give in, which was very reluctantly, I might say. I then took doxycycline, cleared it up, and then I gave blood when I got back, and sure enough, I was Lyme positive. I had the perfect control because I gave blood before I left. I was Lyme negative, so there can be a high false positive rate. So there's no question that these measurements first alerted me that something was abnormal before I knew. I probably would have written it off as jet lag had I not been measuring myself so closely. All right. And then show you, well, we'll jump over this because I see I've gotten later. Anyway, through this first 2 years of monitoring, I've had 4 times when I've had high heart rate. Turns out my skin temperature was up high as well. I learned that later. And I've had 4 periods where this has happened during these first 2 years. So this is outlying heart rate. You can do this with median as well. Outlying skin temperature. Here are the Lyme days. There's 3 here. There's a 4th there. There were other times that were outliers as well. Well, it turns out 2 of those were times when I reported I had a viral infection. I gave CRP measurements, mind you, throughout this whole period, so I know exactly when I'm sick and when I'm not. And there was even a 4th time. Let's see, it's a circle here. It's right there when I was an outlier. We went back and looked at my CRP measurements, and they were just as high as when I have a viral infection. So in fact, every time I got sick, my wearable has flagged it, including once when I didn't even know I was sick. Okay, so we think that these are very capable of finding these sorts of events. So we've written an algorithm we call change of heart. That works for me. It works for now 3 other individuals, one of whom got sick twice, where we can tell the earliest times when infection we think is coming on or when the symptoms are coming on because of elevated heart rate. So for me and one other person, both heart rate and skin temperature elevated, but for 2 other people, the skin temperature didn't change. I think it's how they're wearing the watch. But nonetheless, we can pick these periods up with us. I won't have time to get into details. We look for a rapid delta change, and we for sure can pick things out with a very low false positive in a 2-hour window. I'm sure we can do it even higher. But I think that's early enough to tell before you realize you're getting sick that you are sick. Okay, we can argue, or not argue, discuss why I think that's powerful information. If for no other reason, you might not go into work and infect all your coworkers if that happens to you. I think this is just the tip of the iceberg. So we think we can pick up inflammatory diseases and respiratory diseases with these devices, but we also think we can pick up other chronic diseases. Turns out there's some heart rate parameters. I won't have time to get into this, but they differ between insulin-resistant and insulin-sensitive folks, and we have some other sorts of measurements we're making here that are very, very powerful as well. I think even more powerful than this. So the bottom line is I think these wearable devices will be monitors, if you will, for your physiological health, and they're going to be very powerful when combined with the omics information I mentioned earlier, we believe. So what I see all this going towards is a world where your smartphone is, in fact, your command center for your health. Okay, because you can relay nearly all this information into your smartphone. We don't have it all geared up right. That's something we're working very hard on now. I also think this can all be shared with your physician if they're willing to accept it. That's a cultural shift we'll have to get into. But I think all this can relate to your physician so they don't have to just see 15-second measurements in the doctor's office. They can see your baseline parameters for the last, whatever, several years if needed. So you can really see when people start shifting, maybe useful for TB and some of the things we heard about this morning when people were first getting infected. So I see I'm out of time, so I'll just say that we do think not only is genome sequencing here, these omics profiles are here. I'm sure Lee is going to expand on this more in his talk. We do think that the power of all this is that these are longitudinal profiles. You're always trying to find deviations from people's personal baseline. That's the key to these studies, whether it's wearables, whether it's omics measurements, whether it's what have you. I think everybody does have a different baseline. That's why we need personal measurements, and I do think we're all going to have to take responsibility for our own health. I don't think physicians of the future can understand you the way you can understand you. These are the people in the lab who did the work. I'll just say when you, Brian Pining, Kevin Contraplantages were some of the spearheads for the omics part, Shal, Jesse, and Dennis for the wearables. We have a terrific collaboration with George Weinstock's lab, and Tracy McLaughlin's our clinical collaborator. And we did get funding at least for 3 years from the Human Microbiome Project. And lastly, I'll leave you with a shameless plug for my book that we had come out last year. So if you're interested in the genomics side and maybe some of the other sides, there's even a chapter on wearables in there. Feel free to buy it from Amazon or something. Great Easter present.